Jauhar. Ali is okay, no problem.
Jauhar. Jauhar.
Yes, perfect.
I think Shubhrata. I think Shubhrata is right. Shubhrata.
Yes, Shubho is my nickname. Shubho
Okay.
and Shubho. Right?
Yeah, sometimes I had a friend previously, Shubhroto.
Okay. Okay.
Yeah, but yours is Brata. It ends like
Brother. Yes.
yeah, Shubhroto.
Shuvo is very easy, and we have been routinely using it on the professional platform also.
Okay. I think you guys are doing an excellent job. I think
Thank you so
that's
much, sir.
bringing the science to what we call scientific masses. So that's a credible achievement. And it should grow very strongly, I believe, because earlier this scientific business was done by some seminar the event organizers. In the name of science, they were just harvesting money from the people in terms of registrations and all those things. And this is free science and freely shared across. That's what even CG wants. CG centers are interested to spread science freely without any costing on it. Yeah, and this is exactly why you guys should be congratulated as a team.
Thank you so
Very
much.
good.
Thank you so much.
I have another question regarding the introduction. Do you say NARES and INJA, or do you spell it out like NARES?
I couldn't get your question.
Abbreviation gulo bol rahe hain.
IN is
How do you?
So NARES partners?
Yeah, it is an abbreviation. So if you say
NARES.
it like that, it will be
That's
great.
okay. Spell it out.
NARES,
Okay.
it's called NARES, National Agriculture Research and Extension Systems. NARES.
NARES. And
NARES means like IRRI or any institution is a NARES.
yeah.
It's a generic word to cover all the public institutions.
NARES.
So it
And
means National
INJA?
Agricultural Research and Extension Systems.
Beautiful. NARES, INJA, and MET. Yes? MET.
MET.
So just say MET.
Yeah, I don't know which context. MET is where
Your
it is written.
introduction, sir. It's multiple stress tolerant materials shared with NARES partners, INJUR and MET.
Yeah, MET trials. That means multi-environment trials.
Okay.
Yeah, sorry for that abbreviations. Nowadays, everybody writes abbreviations so quickly. And
NARES, INJA, and MET. Okay. I just want to try and get things right.
Oh, that's great. Good.
Yeah.
Good work.
Anyway, no problem.
This is a practice session. Get it all out.
We haven't gone live yet, have we?
Yeah.
No, we have. Oh, no.
Yes.
We are live now.
Oh. Sorry, everyone.
You're live now? Oh,
Yes, sir, in YouTube.
So
So what did you think about Jacinta? What would you do with that J. Nehru or Jawaharlal Nehru?
Jawaharlal Nehru.
Oh, gosh.
he'll get confused in
I
Jauhar
can say
and Jawahar.
really
then.
slowly, but I don't know. Oh, I've lost it in my Jawaharlal Nehru.
Yeah.
Nehru.
Yeah, that's fine. That's fine.
Jawaharlal
The last name is important.
Nehru.
Yeah.
I'm
Even
gonna
if
have
you
to
don't
say
get the first one, no problem.
Jawaharlal Nehru. Oh, she's laughing at me.
I'm not laughing at you. I'm laughing at Sir's comment at the last. Nehru is it is a bit of political thing. Don't worry, I'm not laughing on you, Jacinta.
No, it's great
no.
that you're trying so hard.
Yeah.
Okay.
Really good.
I'm just try to broadcast and allow the attendees to come in this Zoom platform.
using this as a stand to lift it up.
I missed it.
Take from banana. A wooden page.
Okay, it's 11. Welcome everyone to our Bioengine platform. Welcome everyone. Welcome everyone to our Bioengine platform. This is our 10th webinar in the series, and today we have a great pioneer scientist in hybridized rice research, Dr. Jauhar Ali, and from the IRRI, Philippines. Jacinta will introduce him. Neha will be co-host this Zoom platform and shut it out the questions and the attendees reply. Shoma will help us to interview to Ali said, and I'm request all of you, if you have any question related to today's box, please clearly mention in YouTube chat box and here the question answer box in the Zoom is specific and related to today's work, today's topic only. Those are the younger and young viewers. I'm request all of you don't anxious about your certificate and password. This will be provided at the end of the talk. And I'm assured you all are getting this link and password. So please don't comment in from the very beginning. It's create very difficult to pick up the right question for the talk. So be patient. We will give it to at the end of this talk. I must thankful to Jauhar sir for accepting our invitation. It is very new platform and it's means to a lot to us. I also thanks to IRRI, thanks to their Hybridized platform. They promoted this talk very well. I'm also very thankful to Global Plant Council. They also promoted this talk, and we got so many registrations and so many excited plant scientists and plant biologists. So I request Jacinta to introduce our speaker.
Thanks, Shrivo. Welcome to today's webinar organized by Bioingene. Bioingene is a non-profit organization created to promote plant research worldwide. The seminar series has been designed to build a platform from where plant scientists can present their research to the world. We hope future scientists can gain perspective and inspiration by listening to the esteemed plant researchers talk about their scientific accomplishments and their thoughts on the future of plant science. We are grateful that many renowned scientists have accepted our invitation to share their research insights with us. We have had a huge response of more than 5,000 registrations for today's webinar. We are thankful to the International Rice Research Institute in the Philippines for sharing and promoting today's webinar all over social media. We're also thankful to the Global Plant Council for their encouragement for promoting today's webinar on their platform and social media. We also welcome the audience and thank them for their interest in the Bioengine webinars. It is our request to all viewers to please hold all questions for the question and answer round at the end of the presentation. You can type your questions related to today's talk in the Zoom Q&A box or in the YouTube comment box. You can apply for a certificate of attendance via the feedback link, and the password will be provided after the presentation. Submission for the feedback form is mandatory to receive a certificate. Today's topic for the webinar is Climate Smart Rice Hybrids by Dr. Jauhar Ali, a senior scientist at the International Rice Research Institute in the Philippines. Dr. Ali, a hybrid rice breeder, is a pioneer in his field of research. He has significantly contributed to the development of multiple stress tolerant inbred and hybrid varieties and capacity building for strengthening global agricultural research. He has developed and characterized 8 thermo-sensitive genic male sterile TGMS rice lines, for which he was awarded the Jawaharlal Nehru Award for outstanding PhD thesis research work in 1994. This work led to the discovery of the genetics of the TGMS gene and to the understanding that TG genes, TMS4 and TMS8, were unique and could be exploited. Using these materials led to the development of several TGMS-based hybrids involving different centers. Currently, Dr. Ali has taken up 2-line hybrid rice breeding at IRRI on a strong footing to deliver the 2-line hybrid rice technology in Asia. He has developed several low critical sterility temperature point TGMS lines that are currently in the verge of commercial exploitation. Also, he has been instrumental to the development of the green super rice breeding technology at IRRI, being the project leader and the regional coordinator of the Green Super Rice GSR project in Asia and Eastern and Southern Africa. Through this innovative approach, he has bred and released 26 rice varieties in Asia and nominated nearly 104 GSR cultivars into national cooperative yield trials. He connects genomic resources and tools to breed superior, high-yielding, multiple stress-resistant rice varieties and hybrids. As lead and regional coordinator, Dr. Jauhar Ali leads the development and targeting of improved rice materials through systematic testing, adaptive trials, and varietal release for 8 countries in Asia and 5 in Eastern and Southern Africa. 56 GSR inbreds and 22 hybrids have been released, whereas 110 are nominated for national yield trials in different countries. Dr. Ali has also established the IRRI GSR breeding front in 2009 and produced more than 530 promising multiple stress tolerant materials shared with NARES partners, INJA and MET. He has published more than 80 peer-reviewed publications. He has also provided training to more than 2,100 researchers, especially on hybrid rice technology, seed production, and molecular breeding. I now request Dr. Ali to begin his talk. If you could please share your screen with us. Thank you.
Good day to all of you in whichever time zones you are all. And I would first thank Subroto for organizing this event under the Bioengine platform. with his, and I really, they deserve congratulations for doing such a wonderful task in a global scale, and bringing the science to the scientific masses, and at no cost, especially in this COVID times. I think this is exactly the reason we should congratulate all of us, Shuvo and his team. And with that, let me begin my talk on strengthening global food security through climate smart rice hybrids. If you look at the food security, rather we should look at the food insecurity in the first place in order to do something for the food security. Food insecurity map published recently, where it shows the current levels of food insecurity in many countries in Africa and Asia are in light orange color, which shows the food security insecurities are very high. But the situation still goes worse when the global population would be something like 9.7 billion people in 2050. And if you look at the map at that time, it will be something like a very dark orange red, which is a signal that we are heading towards that. And among them, the conditions under which we are going to raise this, the food insecurity challenge, meet this challenge, we will be under climate change. And among the stresses, if you look at the drought, is picking up like anything. And that means heat and drought would be the major factors that would determine how agriculture would survive. Especially the most affected would be crop plants and the livestock. So this is really a challenge that is coming in the coming days. Now, securing the food under climate change is very, very important. And there are 4 elements, the key elements. There are plenty of them, but I listed here 4 important elements out here. The first one is basically the core ingredient, that is the climate resilient crop varieties and hybrids. This is not only for rice crop, but for any variety, for any crop, across crops, food crops, it is applicable. And where we need to enhance the yield potential under both favorable and unfavorable environments. And the same thing can be realized, these hybrids and these varieties gets well expressed if your crop management technologies are properly associated with it and properly advanced to the farming communities. And the other side of the coin is also, when you talk about food security, we have to talk about the nutritional security as well. And this is basically comes when we have to use biofortification approaches, and especially for food insecure and chronic regions of the world. And above all this, the conducive policies and action plans across the policymakers in different countries to keep the food reachable at all times. And that means the food availability, its access, affordability, its utilization and stability plays a very, very important role in the food security plan. And in this picture, the hybridized technology is a very vital picture. You have to understand the context here. If you have to sustain the higher yields and grain quality, hybrid technology is very, very suited for this kind of situation. And in many countries where food security is a concern, where limited land is there is the best option is to switch to hybrid technology, whether it is for rice or even for other crops as well, where the per unit area productivity would be increased. And coming to the self-sufficiency in rice production, many countries have to keep their bowls filled. At times, when you remember when we had the food crisis in 2008, many countries were suffering from the food were not in the shelves. And many of the countries closed their export gates, including India and countries like Thailand and Vietnam and all these countries. And that's the reason why self-sufficiency in many parts of the world is very important as far as food is concerned. Hybrid technology, when you grow hybrids, it gives about 25 to 30% yield advantage. That means it frees up the land to other crops and other important utilization of the land. The inputs and the irrigation water is saved about 25 to 30% in the same proportion. And the technology not only it generates a lot of rural employment and seed industry. If you take a typical example of Hyderabad, the surrounding areas of Hyderabad, where the hybrid technology is one of the hot centers in India, you see the rural employment in those villages. If you go around, people are well versed how to do seed production, and they are very good in managing those technologies very well. Now, coming to the less dependence on rice imports, many of the countries, if they have to reduce their dependence on the imports, then the best option again would be hybrid technology. And especially when you compare with wheat, wheat has surplus of wheat available in the international market. But if you look at the rice, it's somewhere between, it fluctuates between 25 million tons to 40 million tons in the international market. And countries like China and India, whenever there is any crisis, they will mop up all this 25 to 30 million tons in no time, and leaving many countries without any food. So in the coming days, this phenomena would really would be of major concern. So people at different levels, at different countries, they have to look how they can become less dependent on the rice imports. And of course, it nurtures the seed industry to produce quality seeds, not only for rice, but also other crops. And the quality seeds is the best method to deliver the good productivity and increased production in any country. Now, if you look at this map, the graph, you see the area outside hybridized spreading across outside China is almost now 8 million hectares outside China. And this is the potential of this growth in South Asia would go up to 15 million hectares, whereas in South Asia, especially Myanmar, would be around 5 million hectares. And the current area in China is around 16 million hectares. So this is the trajectory that we would expect in the coming days in order to feed to meet the requirement of the extra food that is required as far as rice is concerned. Now, if you look at the core research challenges for hybrid breeding, the first and foremost is increasing the grain yield heterosis. Can we increase the yield heterosis beyond 25%? That would attract the stability, that kind of stability would attract the farmers to adopt hybridized technology. Then the question comes is, can we breed multi-stress breeding for developing the parental lines that would match the market needs in the region? And coming to the consumer and grain quality for the market of the target regions is very, very important. Many times we always associate grain quality with, especially of the hybrids, in relation to the Chinese food requirements, where they prefer sticky rices. So immediately the connotation goes that hybrids are sticky. It's not so. Basmati hybrids have been created and they have been catered to the needs. So we can cater to any particular need, any kind of quality is possible through hybrid technology. And then if you look at the new technologies that are coming with the precision breeding approaches, utilizing the informatics on genomics, high throughput, phenotyping, and environments, all this can be combined to create the precision breeding for hybrids as well. And the genomic predictions and artificial intelligence, and to identify heterotic combinations by this approach is feasible now. It's not in the past that it were not feasible, but now it's very feasible. And many countries are looking at this as a big opportunity out here. And the high outcrossing trait breeding, especially, would increase the seed reproducibility. So including IRRI, we are working on this particular topic. And this is one of the biggest challenges. If we can increase the hybrid seed reproducibility beyond 3 tons per hectare, that means we can bring down the cost of the seed so that the farmers can adopt at lower cost of seeds. Coming to the reducing cost of hybrid seeds, we can go to another level of introducing the two-line hybrid technology, where the heterosis per se also increases. And the key element out here is, can we get low CSP or low critical sterility point temperature point parental lines below 24 degrees centigrade. Now, to give a context to this, what is the current levels of the yield that we saw in our multi-replicated trials, MRYT trials in the HRDC platform? We see that in recent times, we see the increased the heterosis over the best check PSBRC 82. We see about 30%. And this is exactly we saw in our, even in our actual trials on the socioeconomic trials conducted in the Philippines. We see the similar number of 30% advantage of the hybrids over inbreds. And I know that many would appreciate that this is the current level is between 25 to 30% yield advantage of the hybrids over inbreds. Now, looking at the different market segments, and we see the South Asia, Bangladesh, and India, and we see the medium slender grain segment and mid-early segment, and in India, mid-early segment. And you see the potential area that is that you see about 8 million hectares. And the yield advantages that is required in this, if you look at the benchmark, is around 8 to 10% over the existing best hybrid. And over the Czech hybrids is about, again, 20. But if we are targeting around 25 to 30% in our screening approach. And if you look at the key gaps that are presently in Indian context, where lack of stress-tolerant materials, producibility, the seed reproducibility, lodging, and false smut are some of the key traits. Likewise, in Bangladesh, the cold and disease resistance, including drought and salinity tolerances. So the same market needs that are required in Philippines and Indonesia is also very important. Like in Indonesia, where the BPH with rugged stent virus and BLB and blast and drought are very important to close the gaps. And likewise for Philippines, we have multiple abiotic and biotic stress tolerances is required. And much of this is also on the grain quality, BLB, BPH, and low head rise recovery are some of the key factors. In this context, what climate smart rice hybrids could deliver. So this is a concept again, in order to create this type of hybrids that would have high and stable yields. At the same time, provide the abiotic stress tolerances, multiple abiotic stress tolerances, including drought, salinity, submergence, and the biotic stress tolerances like BPH, BLB, blast, major biotic stresses. And it should match the grain quality features of the different market segments and market checks, and amenable to direct seeded as well as transplanted conditions. Why I refer here on the direct seeded conditions is in the future, we'll see that the direct seeding would become a kind of a trend because of the lack of availability of the irrigation water, especially creating terminal droughts even in irrigated areas. And this type of direct seeded conditions would increase in the coming days. And what are those things will be covered even in this talk. And high resource use efficiency is another important trait. If you look at the phosphorus and these, especially phosphorus is basically from the rock phosphates, these are going to deplete as we see, as we go along, coming towards 2050. Much of these are depleting resources. And many of them are petroleum-based resources where nitrogen, fertilizers, and all these are going to deplete as we are talking. And we need a resource use efficiency on the prime topic that has to be in these hybrids as well. And hybridized reproducibility of more than 3 tons per hectare is very, very vital for us. And meeting the market requirements of the target region is the core essence of this strategy. Now, in doing so, we need to understand what is the breeding, how we breed the parental lines for these future climatic conditions. And among the major conditions that I look from a research perspective, the drought and heat tolerance would be very, very vital, and these would directly affect many of these floral traits. Now, if you look at the floral traits that the CMS lines should possess, is the long stigmatic surfaces and high seed setting rates, promoting more outcrossing traits. And likewise, the restorer should have heavy pollen load, and dehiscence duration should be able to cover the female parent. And the climate resilient parents with ideal seed production traits, especially higher combining ability, disease resistance, grain quality matching requirement of the market requirements. All these are would lead to eventual adoption or wide adoption of the hybrids. I would like to share one very important slide here. This is which I've shown in many of my presentations in the past. This is a very classic slide where this experiment was conducted in Bohol in the Philippines, where this farmer Apolinar Asa in Kandahar region 7 planted this, we call this line GSRIR 1-8 S6 S3 Y2. It's also called NSIC RC 480 now. And this guy planted this before its release even in this region where the seawater inundates immediately once you when the nursery is transplanted, and in 4 days after transplanting, he sees the seawater comes in because this is near to the coastline. And after some days, he sees the freshwater inundation of the heavy flooding causes 4 days of complete submergence of 1 meter head over the plant height. And then the plant recovers in the same place, and you'll see the plant recovery in the same place. And in the same place, then drought strikes quickly in this place, and where you'll see the drought cracks in the field. And at flowering, you'll see the crop. It looks very good, and the farmer is able to harvest in these circumstances where 3 major complex traits, salinity followed by flooding and drought. The farmer could harvest 1.2 tons when he never harvested anything. In fact, the farmer was smiling when the seawater came in. He was so sure that nothing would survive. And this is what happened was, generally the farmer would not get any yield. Many of our GSR also were without any harvest in that particular, when we tried 5 of the GSR varieties were planted in that situation. The most interesting thing is what happens in the normal, when the normal things prevailed, the good season came, then the variety shot back to 7.23 tons. And this shift is the very important reason why the Department of Agriculture in the Philippines took this very seriously and increased the adoption area of this particular act. Today, it has been released as NSIC RC 480. It was released in 2 conditions simultaneously, in salinity and rain-fed drought. And therefore, it was named as GSR 8, and it now occupies 0.7 million hectares in the Philippines and becomes the first truly climate-smart rice across the crops. And this particular variety does very well in transplanted condition. It does very well in the direct seeded conditions. It has very good cooking quality, and it looks like this in a normal crop at IRRI. And it has the good cooking quality traits and very good in the saline and rain-fed environments. And it's spreading into the irrigated areas as well. We did a survey based on our socioeconomic scientist from UPLB, Professor Yorobe, who is a lead economist there. And based on his estimations, he found that nearly 1 million hectare has been covered in the Philippines alone. And this particular variety touched about 0.7 million hectares. And it's also very good for including iron toxicity areas. So the reason why I try to show this is basically to give you the excitement how this the technology behind it is also illustrated here. What went behind in breeding that particular variety, one has to understand. And this is a very important breeding strategy. We call it as GSR breeding strategy. And it was very, very developed at IRRI, and it was evolved with the time tested over many, many generations over the last 10 years. And what happens here is we use the BC1F2 generation. You make a cross with one recipient, highly adapted genotype, and 10 to 15 restorer donor parents are crossed, and you create BC1F1 and BC1F2 populations, bulk populations. These bulk populations are simultaneously screened for multiple abiotic and biotic stresses. And this BC1F2 is again progeny tested in BC1F3, and then again third round of testing to confirmation in BC1F4. When you do 3 rounds, you are comparing it with the best checks, the trait checks, and the best yielding checks, and this is all carried out also on the normal irrigated conditions as well. So we use these materials around 1,500 introgression lines, selective introgression lines are created, which can be utilized for mapping, trait mapping for any of the traits that you had screened, and it easily maps them, and then you can use the best yielding ones, about 120, to create the preliminary yield trials, replicated trials basically, and then advanced yield trial also replicated in dry and wet season, 2 seasons, to reduce the number to 60 from 120. And then this is sent to the multi-environment trials in different countries, and the partners in 16 countries tested them, and then they nominate into their national trials, and eventually these varieties are released to the farmers. Now, if any of these introgression lines coming from 2 different donor parents, but a common recipient parent, can be used to make a cross, that is called the design QTL parameting approach, where you know a particular set of traits coming in one introgression line, but coming from a different donor line, but both share the same recipient parent. And these 2 are selectively crossed based on the molecular data. And in F2, again, this is screened for 2 rounds, because in 2 rounds, you can fix the homozygosity, and it can go into the trials. This is the advantage of this approach. And by this approach, both from the Chinese Academy of Agricultural Sciences, that shared the GSR varieties in their breeding systems, as well as the IRRI's main breeding program, GSR breeding, we could develop about 28 of the varieties were released from IRRI's program, and in total 56 were released. And the last release is in India, where IIR 56 done, which is very important in the context, because it is being released in Punjab, Haryana region. And Huang Wangzan PR 126 is the recipient parent of this particular line. So PR 126 is already covering 30% of the area in Punjab, and because of its duration is 15 days earlier than PUSA 44, that makes the less irrigation water is required, lesser chemicals, lesser pesticides, and it makes it very much fitting in the rice potato cropping system. And that way, this particular line can easily replace it, because it has drought tolerance and salinity tolerance. In addition to the yield levels are much impressive than the first generation of GSR varieties. So this gives us a lot of hope, and there are more than hundreds of these type of materials in India nominated in the ACRIP trials. Now, if you look at the 28 of them is directly developed and at IRRI and tested, adapted, released, and in target countries. The record time was 7 to 9 years of time, and 2.7 million hectares currently deployed. 110 climate smart varieties from IRRI are in the pipeline for release over the 6 countries. And to have a look at the closer look at these materials, how they perform under a normal condition and in low input condition, under rain-fed conditions, and in what was the relative advantage of across different conditions. We see that many of the traits is more than 10% up to 30%. You can see the advantage, and these are the checks below, and with which it is compared with the target traits that are used, including an hybrid SL8. So what we realized that to adopt this into the hybrid breeding program, we can create a similar set of recipient parent from a particular heterotic pool, and creating the donor pool of 20 restorers, we can create a similar set of approach where you screen multiple stresses in 3 rounds, and then create a set of elite good combining ability backgrounds in the adaptable recipient parent, and this restorers coming from the same heterotic pool. We can create hundreds of these restorers with climatic suitable for climatic conditions, and especially multiple abiotic and biotic stress tolerances. So this is that approach. This can be extended to even to the maintainer breeding programs. So by this way, we also use the what we call some of the trials that we created under irrigated, low input and cold and salinity. You can see the advantage of the materials. And in our stress in drought is so severe that it kills the trial even. We give about, after 30 days of irrigation, after transplanting, we retract the irrigation in the drought plots, creating more than, after 51 days, there is no irrigation water in that field. And that even kills much of the material. So that is how we screen drought. And when you want to combine, I told you that when you have the common recipient parent, like this particular cross had the common recipient parent Huang Wang Zhan, and this was coming from one parent, and this is coming from another parent, but the same recipient parent. We can combine the restorer genes, both are having restorer genes, and then we screen them in different screens, and these selected ones are again redistributed to multiple screens. And after that, we can even do a resequencing or GPS genotyping of this material. And based on this, we can create the best materials that can be released as inbreds, as well as it can be used in the hybrid breeding program. Now, some of these DQ lines, if you look at the design QTL parameting lines, were much stronger and much higher yielding than the parental lines that we developed through the regular approach of interpretation breeding. So design QTL parameting gave better yields under irrigated. It touches something like 10.4 tons. And to just look at this type of materials is really a wonderful sight to see these materials in the field. And we used the GBS called Turnable GPS from data to bio. And this publication was done in Frontiers, and you can follow this. Just to give you an illustration, after the BC1 F5, you can see most of these loci are either homozygous or the major homozygous or minor homozygous. You don't see heterozygous loci at all. So the fixation of the genotypes is within very rapidly done in 3 rounds of screening, and that makes it to achieve the genetic gains much faster than expected by normal approach. So these severe stresses causes the rapid fixation. And this process, when we try to analyze the non-synonymous synapse in these materials, and these Sarkos diagrams can easily show you that most of the color-coded green ones are the biotic stresses, and the red ones are the abiotic stress tolerances, and the blue ones are the other stress-related families. You can see when we screen this material for these major genes, known genes, you can see many of these populations, the interrogation lines, had these genes selected by this approach. So now we can easily know how to parameter these interrogation lines by design, and this is how this whole technology works. Not only that, we can also map different target traits for different target rates for drought, salinity, and we can easily get these peaks and with high Lord values. And these have been mapped very well in many of the populations that we studied. Now, to cut this story short, many of these multi-stress now were mostly in the restorer backgrounds. And that made it easy for us when I took charge in 2016, on the hybridized. Immediately we initiated the crosses between the stress tolerant with the CMS lines and the TGMS lines that we developed, and made these crosses. And we saw very easily that much of the heterosis was scaled up beyond 20%. And in two-line hybrids, it even touched 40%. And this is a very important beginning for developing the climate smart hybrids. And today we have the first generation of hybrids ready, and it is going to be tested in different parts of the world. And the second generation would emerge when we convert the CMS line also into climate smartness traits into them. And then when both sides, both the parents have these traits, that will be the ultimate objective of our breeding program, to develop the climate smart. So basically, to summarize it, the genomics assisted hybridized parental line breeding, what we did was basically the simultaneous multiple abiotic and biotic stress screening selection scheme is a very interesting approach by where we can employ early generation backcross breeding approach to screen for restorers and maintainer backgrounds by this innovative GSR breeding strategy, where we can not only utilize for QTL discovery, also for the hybridized development. Coming to the several promising parental line developed with multiple abiotic stresses are now currently used in the climate smart hybrid combinations. And the genomic assisted breeding also help us in understanding which crosses to be used for the design QTL parameting approach, and pyramided and stacked more genes for abiotic stresses over each other. And then we can also pyramid non-allelic QTLs or segments that can combine many of these traits from different donors into common recipient parent to develop the climate resilient hybrids as per design and needs. So at IRRI, what we are doing currently is we are trying to create heterotic pools, and that has been now created. And we use the 3K genomes that was used in our breeding program, where more than 500 of these materials were used for directly in the multi-stress breeding programs. And now we have classified our source nursery into different heterotic pools, as you see on this side. And now when you cross the wider genetic pools, very likelihood that you will get good combinations. And then we are employing the artificial intelligence technologies and also genomic prediction models and machine learning tools to predict the best combinations and to reduce the cost in identifying the best combinations. And this way we can be sure of our success rate increases. Recently, we published a paper on the heterosis breeding via genomic selection in rice. And this paper was published with the UC Riverside, Shizhong Xu, who is the scientist at UC Riverside. And the first author is a postdoc, and she did this work. And this is a wonderful work where they used the existing rice population of 1,495 and using GBLUPs to perform the hybrid performance predictions. They could also not only replicate 10-fold validations across prediction abilities on 10 agronomic traits that range between 0.35 to 0.92. But if you look at the yield prediction was 0.54, but major genes like grain length was 0.92. And now you can predict using these founder lines, you can predict even cross combinations without need of your parental lines to be in that set of founder lines. This would be extended to the current IRRI breeding programs, where we'll be extrapolating that information to predict the best combinations here. And the other scheme that I would like to highlight here is the 1000 F1 trial. This 1000 F1 trial is a very, very ambitious project, which is already started in 2019. And there are 10 locations basically. We don't cover 1000 F1 in all of the sites, but we divide these 1000 F1s into 50% coming from the MRYT trials that the HRDC members contribute to the HRDC membership. They have their slots of known hybrids with their best performing hybrids from the industry. And 50% of the hybrids are from the IRRI hybrid program. And 20% of this, so let me put it like this, 40 coming from private sector, 40 coming from IRRI, and 20% coming from the best market checks, including the best market checks in their locations. And every 40 of these individual hybrids are basically customized for each location, so that the people who are evaluating, they are looking at the IRRI hybrids that are customized for that region and comparing it with the best hybrids from the industry and best check hybrids in that region. So basically, this is called a kind of spatial testing and sparse testing. And you can see the grain yield performance of all hybrids in each location is predicted. The best hybrid combinations in different and across regions can be identified. And each cooperator would know which hybrid combinations are doing well. But at the backside of this, we know at IRRI, we'll know that which of our combinations and which parental lines did good in a given target region. And immediately these breeding materials would be quickly employed to create the next generation of parental line breeding. We'll rapidly go by the RGA, rapid generation advancement, and forward breeding approaches to breed superior parents. And these superior parents would help in developing the superior hybrids. And in this way, a cycle of another set of 1000 F1s would be created. And this would help in achieving the genetic gains in the hybrid breeding program. And this is very important from a hybrid research standpoint of view. When your hybrids get into multiple environments, and this will be super laid over by grain quality and disease, and other layers will be layered over it. And this would increase our collective genotyping and phenotyping approaches that will be helping us to map only all the major traits that are concerning the hybridized research. And this will be a fantastic design, but to implement and get it done in the coming years is going to be one of the biggest challenge, especially in the COVID times when seed movements are highly restricted and taking more time. And this is one such plot at IRRI of the 1000 F1 trial. You can see this was in 2019, and we saw this results were very good. And we also not only working on the very important insect BPH, which is much influenced by the climatic conditions, and they come in big waves especially in Indonesia, where BPH is one of the major problems there. And this BPH38 is a novel gene identified in the Huangguangzhan background with gene coming from the Khazar donor. And one of the reasons for this, we narrowed down to this particular 260 loci, which could be one of the F1 F-box proteins and possessing the leucine-rich repeat domain, and could be involved in the salicyclic signaling pathways that gives BPH resistance. And we are trying to incorporate this into our breeding materials, especially restorers and maintainers, to confer this tolerance into the hybrid as well. Also, at the same time, we are also working on the nutrient use efficient traits. We mapped recently a paper which was published in PLOS One. You can refer to that. But to cut this story short, we could map 19 QTLs that were screened at different stages, at the same time at different levels of for the nutrient used, like 0 NPK, 80% NPK, suboptimal, and at 100% NPK. By doing so, we could easily detect two major hotspots, especially on chromosome 2, especially the OSNPF 7.1 and the OSGF4, And this is a growth regulatory factor, and the NPF is a nitrate tripeptide transporter. These technically helps in the use efficiency pathways. And what we achieved in the hybridized program is we could successfully release 17 hybrids, out of which 6 were first time commercialized to the private sector on limited exclusivity for a 6-year period, to especially 3 of these hybrids to Tau Corporation and another 2 hybrids to SL Agritech. And we have currently members up to 88 in the HRDC membership. We publish our annual report too, and it's shared with the HRDC members annually. And many of the platinum, green, and green members are free to join. There is no cost for them. The private membership starts at $20,000 per annum, and the platinum starts at $45K. And these are the models by which one can really utilize the germplasm that is freely shared with the public sector, but it is given to the public and private partners for their members, for their membership. And we bring this value to them. These are the 6 hybrids that I mentioned. One of them is a 2-line hybrid, MST-61. I take you to another topic where, which is much related to the direct-seeded hybrids. We are working very seriously on this topic on development of direct-seeded hybrids. Our first low-hanging fruit was Mestizo 89, which turned out to be very, very good for direct-seeded conditions. And this was verified by the Direct-Seeded Rice Consortium, which is a consortium for direct-seeded and led by Dr. Virender at IRRI. And this is one of the early situation where we could identify this. Now we are having a very full-fledged breeding program on the parental line breeding, and also developing many DSR hybrids. And using the introgression lines that we developed in the past, many of them were also direct-seeded high inbreds that we developed. And these are now in the restorer backgrounds and directly utilized. But to understand the key traits that are required to develop the DSR hybrids, the foremost is the anaerobic germination tolerance, emergence from deeper soil depths, shorter duration, high yield potential, weed suppressiveness, and herbicide tolerance should be incorporated. Lodging resistance is a key trait, and tolerance to abiotic stresses, especially drought and nutrient use efficiency, are very important. Provided we have this, these type of direct-seeded hybrids can be also, we can employ alternate wetting and drying strategy, thereby reducing the greenhouse gas emissions in these plots. And direct-seeded rice is really the future in the coming days. And we also introgressing BPH, Tungro and Blast and BLB resistance genes into the maintainer and restorer lines, elite restorers and maintainers, which will be shared with the HRDC members soon. And also IRRI has been in the forefront to share such knowledge of marker-assisted breeding for hybridized technology. And we have listed here several of these trait-based, gene-based markers, which are available for people to use for the marker-assisted breeding and utilization in their pipelines. And also, IRRI introduced the forward breeding approach for hybridized with 10 markers with the Intertech panel, which is also available for people to use for their breeding efforts. And by this way, we can do a forward breeding approach where many of the B by B and R by R crosses can be filtered quickly in the early generations for using these markers, and progress in RGA single seed descent approach rapidly to F3 to F6 in 1.2 years. And therefore we can create materials in 2 years that can be shared with the members. I try to cover another important area which everybody would be excited is the 2-line hybridized technology, how this helps in the climate smart hybrid, climate smart hybrids to propagate in much cheaper and much sophisticated manner. But now to understand this, one has to understand temperature-sensitive genetic male sterility system for two-line hybrid development. Now, this is a major gene, and it's a recessive gene located in the nucleus, and it requires low temperature to become fertile, or and this can be achieved in the low temperature conditions where the plants are raised and multiplied. And these seeds are brought to high temperature regions, and where it can be restored by any non-TGMS, which we call as pollen parent, can restore the fertility of this. So there is no such thing like a restorer here or a maintainer here. Any parent can restore the fertility in the TGMS lines. And this is how the benefit is: you cut away the maintenance part, and the cost of seed production can be reduced by at least 40 to 50%. And if you see the history of the two-line technology, was way back when I did my PhD. We developed 8 TGMS lines, and many of the early findings are there in this manual that we published long back. But this technology never lifted up, and we understood that one of the major reasons, despite the advantages that it has, like I told you, maintainer can be avoided, choice of parents are broadened, and the genes are major genes, so it can be easily transferred. There is no negative effect of the cytoplasm that the hybrids do have. If you have the WS wild abortive cytoplasm, it has also higher seed production ability than the 3-line system. So we can easily cross 3 tons through this approach. And 2-line hybridized generally yields higher than the 3-line hybrids by 10%. So that makes it attractive. Now the key element here is to produce a hybrid parental line that will become sterile, can become completely male sterile at 24 degrees centigrade or even lower than that. So this is our objective here, to breed this. And this was not available in the past. And recently, over the last few years, we could develop this particular line. And we have created a new study group called the 2-line study group, where a set of like-minded companies have come forward to fund this research. And we have been successful in going forward in testing and validating for commercialization of the 2-line in India and other parts of the world. And if you look at one of the 2-line hybrids here, 554, which was tested in Hyderabad in 2018, and we can see the advantage over US 337 and MTU 1010 by 21%. And this is another old TGMS line using the older version, which is also doing good and did well in Hyderabad. And we had also done in Varanasi last year in 2019, where it outyielded IRRI 6444 gold by almost 7%, and this 554 did with 6.5%, and over SAVA 127, 13, and 12%. So this is very important to understand that the 2-line hybrids are important features that can lead the way based on especially reducing the cost of seeds and the heterosis, increased heterosis. 554 has a climate smart traits like drought and low input, or we can call it as nutrient use efficiency is higher in these hybrids. And these are being validated and tested in the different sites in India now. And I'm very optimistic about this hybrid's performance, which is already nominated in the Philippines National Yield Cooperative Trials. Now, to summarize the whole talk, what would be the future hybridized research direction? Now, climate smart hybrids are being evaluated now in vulnerable target regions in different parts of the world. Newer climate smart products involving heat, cold, elevated levels of CO2 are the key areas, including even ozone. These could be one of the few futuristic climatic conditions would warrant us to work on these ones, including nutrient and water use efficiency. So the hybridized program currently is dealing with these areas for our research. And we are trying to look at the interactions that could be also interplays would be there between these target traits, and how these could be pooled properly by good breeding design, And that's what we are looking at. And designing new CMS lines and TGMS lines that matches the market requirements of 3-line and 2-line hybrids are very important. And innovative approaches like genomic prediction of heterosis and artificial intelligence could narrow down our combinations that we would like to test, and cost reduction and precision heterosis breeding is going to be the future. 2-line hybridized technology would become a commercial reality in tropical Asia, provided we have the usable low temperature or the low critical sterility point below 24 degrees centigrade. If you have something like 23 or 22, I'm sure we will be able to get it right. And the genomic editing tools, I don't know, many countries still are keeping this under the GM category, but sooner or later, this would be understood as one of the future techniques, and it would become as a routine. And it will be easy for us to edit target traits and quickly get to the help for the farmers in addressing some of the key target traits correction, and we will be in a better situation in this regard. And hybridized technology cannot be lifted without the human resources being trained. And this is one of the very key aspects where we are very much in the HRDC as well as other platforms. We are very much concerned for training more human resources that can understand how to breed climate smartness in their parental lines, and how to develop hybrids, and how to do seed production in their own respective regions is very important. And IRRI also has we would like to look at the future to create a center of excellence for hybridized research and extension that would cater these type of things in the future. And this is something which is very much practical, keeping the consideration of food security. I think wise investment should be in this direction to create this type of centers of excellence for such an important technology. And of course, socioeconomic impacts should be tested, should be tried for the climate smart hybrids in the coming years, especially in Asia and Africa. I think I will try to conclude here, and I would like to acknowledge all my team members, especially in the GSR project that was funded by Bill and Melinda Gates team, and the entire GSR team that participated from CAS, Chinese Academy of Agricultural Sciences, IRRI, and Africa Rice. And NARIS partners from 16 countries are all duly acknowledged. We have our rice breeding platform leader, Hans, who has helped us to project this whole new dimension of the hybrid breeding platform, hybrid breeding cluster in a different way. And also, I like to acknowledge Remy's contribution to take it to the private sector in a very energetic manner, and his team also comprised of Ajay, Linga, Roberto, and so my team from Anna, Pauline, Tini, and also the HRDC Advisory Committee, hybridized breeding team involving Lito, Kaloy, Eric, Anna, Christian, Donna, Roy, Bea, Jonas, Lolit, Makto, and Sinan. I really appreciate their help and in making this possible. And our breeding clusters approach is written in this caption here: developing superior high-yielding heterotic rice hybrids that meet market requirements, adopting genomic tools, and to strengthen and invigorate the hybrid seed industry through the Hybridized Development Consortium by providing elite hybrids and parental lines and technical know-how. With this, you can visit our websites, HRDC website, for more information. And IRRI's 60th year, I think we are marching forward to go beyond rice. Thank you very much.
Thank you, sir. We have some question in Q&A box. That is after the screen share, the polling, and then Q&A.
I'm not able to open that. Just a minute.
In bottom.
I have to stop. I think stop sharing.
Yes.
It, right?
Okay. Sure. Yes.
Okay. Now it comes actually. Okay. Good.
Yes.
Okay, so
we put some of selected them, but comes randomly, so you can answer few of them from that Q&A box section.
where should I find that?
It is there.
Okay, great. Okay.
Yes.
Okay.
34. It's source 34.
34 questions. My goodness. Okay.
You are interesting. You are interesting.
Yeah, it means a lot of interest it has generated. Anyway, the hybridized varieties released in India are yet to release. What are the varieties and what rates and yield rate, tons per hectare? Is it successful in India or not? I think you should understand that ICR, Indian Council of Agricultural Research, with its partners, especially IIRR and IRRI and NR, National Rice Research Institute, are members of the HRDC, and we are sharing with about 26 private members from India alone, who have utilized our materials, and not only our parental lines and materials. Also, we are sharing with them many of the varieties from the HRDC, which they have been testing. And I'm sure many of them would be in their coming to their pipeline for release and utilization. As of now, we have not any major release in India in terms of direct release, but in the Philippines, we have made 17 releases. I think I will go to the next question. Yes. Can you explain how artificial intelligence predict heterotic combinations? Yeah, these type of tools are basically coming from some outsourcing done by Computomics, and other agencies are there which can use artificial intelligence to predict the hybrid combinations. Basically, this uses the genomic knowledge. The genomic sequences are known. The environments in which they are to be grown, this is known, and their phenotypic traits are known for a given set. And then in the places where they are grown, once that of the founder lines are known, then it's just like putting more and more data into it. It starts, it's a machine learning process, including the GBLUPs, and the BLUPs would kind of machine learning process. And the artificial intelligence basically breeds on this and accumulates more and more information as it becomes more intelligent by itself. And it will say, okay, use these combinations. These are the best ones for you. I think this is the way it does the artificial intelligence, but it needs a lot of information in the first place coming from different trials. And this gets fed into it repeatedly by very good algorithms of machine learning. And that's how the artificial intelligence grows. It grows with time, actually. And then we have a grain number per panicles in hybridized varieties. Is it 300, 400? It is not important what is the grain number. It is ultimately the grain yield. Sometimes there are compensation mechanisms. In some panicles would have more, something like 350 to 400, or some would have 250 to 300 grains per panicle. But then the number of tillers, number of productive panicles, and the sterility at the bottom of these spikeletts, all this matters in the total grain yield. And I would suggest watch out for the final grain yield per plot. That would determine ultimately the yield. And of course, these are yield component rates. It matters a lot, but higher with more productive tillers is very essential. The next question is from David Chen. Sorry to clarify, what are the desirable traits and characteristics of variety that makes it better for direct seeding, in case I was unclear in the previous question? I think the direct seeded conditions, basically the traits that are important. You see, when people will throw direct seeded in dry bed condition or wet bed conditions, basically in wet bed conditions, you need anaerobic germination tolerance, because the moment you throw the seeds, you will inundate with water, and the water would not allow the germination of these seeds, but it will control the weeds. So for control of the weeds, you need to keep the water film over the surface of so that you can control the weeds. And therefore, anaerobic germination tolerance is very vital. then the under deeper depths of soil, especially in dry bed, sowing, sometimes the seed falls toward lower depths, and that ability to grow from that from 2 centimeter below depth, it should be possible for these. This should be a trait as such. And likewise, the herbicide tolerant resistance and I think many of these traits are very important. Early vigor, and if you had seen that figure on that, I had illustrated most of these traits there. I think you can look at that. And herbicide resistance tolerance, like what we are working with the BSF, Clearfield and Provisia technologies are there. Also, herbicide genes can be incorporated that will take care of the herbicides that we can apply on these plants to make it really effective. Another one from anonymous attendee: burden on farmer for seed every season as hybrids segregate. I think one has to understand hybrids have to be used only one season for its exploitation, F1. Once a farmer buys the seed, they have to use one season and not to grow the F2. And the farmer benefits from the higher yields of more than 1.5 to 2 tons in conditions, especially in favorable rain-fed conditions. The advantage is sometimes even more than 50%. And in irrigated conditions, it touches 30%. So the extra yield that he gets, it is possible for a farmer to invest on it. And we have seen this in time again in the rain-fed areas in India. The hybrids are prolifying in these regions where the farmers are really poor, but now their income has increased because of the hybrid technology, where the farmers are gaining on the advantage of the hybrids over the inbreds. And in fact, it is not a burden when you see the profitability in the front. So that is something people always feel that is the way. But I would suggest people to look at the advantage. And the seed rate is only 16 kg per hectare. And we are trying to go below that even. So that is not, when you compare with inbreds, which is 35 kg per hectare, sometimes 150 kg in direct seeded conditions, people are growing. And when you compare this with 16 kg, it should not be difficult. Next question I see is from Afsana Ansari. What is the highest seed yield of hybridized in the world, both in CMS yield and F1? I think the best yields can touch up to 6 tons in the best yields. I'm telling you, it can touch even 6 tons in cases where China has reported. But to be realistic, on an average, you can touch 3.5 tons in China. They can do it. In India, the average seed yields are touching 2.5 tons comfortably, and we are viewing whether we can cross the 3 tons in the tropical regions. So this is very much influenced by the temperature and the climatic conditions where you produce the seed. So one has to understand that CMS yields are largely dependent on the type of CMS lines that you use. So outcrossing traits facilitate the higher seed yields. And CMS yields also becomes much higher than the A/R seeds to the same level, and even in fact more because of the synchronous flowering, and it is more than 3.5 tons we can expect in case of A/B in Indian conditions. And how can we get these hybrid rice varieties in Pakistan? We have a partner in Pakistan, the Rice Research Institute in Kalashakaku is getting it is a member of the HRDC, and the private sector who are interested from Pakistan, they can become members of the HRDC as well, and they can at $20,000 per year, but they can really make money when the time comes for licensing. You have to pay a very low upfront, licensing fees, which is just like 2.5% of a given hybrid, and 1.5% for restorer. and this is very, very low level of licensing fees, because IRRI wants to use these funds not for increasing our coffers, but we want to increase the investment into hybridized research. And this money goes back to the public's utility, where we bring and plow back this money and bring more rich dividends to the hybridized research. And this is how this whole pattern works. And therefore, the public sector receives the seeds free. The private sector receives for token money of or licensing this at a later stage. So I would encourage all private sector, whether it's from Pakistan, India, or any other country who are interested for similar questions, they should contact HRDC Secretariat. We have a HRDC website. I would encourage people to become members and take the benefit to their country and to their region. And I told you, public sector are free to become members. Next question coming from Anuj Kumar. Sir, what is the future of hybridized in India? And now majority of the area is under pure line varieties. I think, Anuj, you are not aware that currently the area under hybridized is 3.5 million hectares. And this area is likely to grow very rapidly in the coming days. When the food crisis is going to hit, people will switch the button to hybridized, because that is the only viable approach any planner would look at it. You don't have any other alternative approach at this moment that can give 25 to 30% increased productivity. And I really, people should align themselves with this concept in their mind. What is the next question, Miraj? What is the difference between hybridized varieties and improved varieties? The major difference is the hybrids have early vigor, and they can be combined with any type of tolerances by complementing the traits in two parents, and you bring it into the hybrid. The grain quality can be customized based on the grain quality parameters of the two parents. So you can really address the hybrid varieties as we want. So it is what is in the parents is what is the hybrid is all about. So you want to have good quality hybrids, you can have good quality, you can have basmati hybrids for that matter. But the bigger difference in improved varieties is 25 to 30% yield advantage is the biggest factor that helps the hybrid varieties adoption. And then Supraja requesting the best method for development of hybrids. I think I showed you all the methods, but IRRI is now engaging on the one rice breeding approach based on the genomic selection. And we are also employing the genomic selection in the heterotic pools and for the parental line breeding. And this is one rice breeding is very, very essential, and will become the future way of breeding, both for inbreds and hybrids. And many countries, and including the CG centers, are already adapting to this one rice breeding. So this is a new area which will be soon, you will be listening to it to more. Okay, so next question is Acharya wants to know restorer line or maintenance line are from wild variety or cultivated type. The gene that came from the sterility gene that is located in the mitochondria comes from wild abortive cytoplasm, which is coming from Oryza sativa spontanea, which was the first source of WA cytoplasm, which was discovered in China, and later it was shared to IRRI, and from IRRI to all the Asian countries, these lines were developed and into the CMS backgrounds, and they were freely shared with the region. So the restorer genes are available in indica backgrounds, plenty of it, but in japonica you don't have restorers, and that is the reason why two-line approach is very good approach to create interspecific hybrids between indica and japonica. Now coming to Raj Kumar, how non-allelic QTL pyramiding have advantage over allelic QTL pyramiding? Under what circumstances we can sort non-allelic QTL pyramiding? I think this is the advantages are relative between non-allelic and allelic pyramiding, but the key factor lies on what type of traits that we are moving and how we are preparing the alignment and the benefits of that. So generally, when you are using different donors and common recipient parents, and these non-allelic segments for a given target trait contributes to the drought tolerance or salinity tolerance, and whether it's allelic or non-allelic. But generally, non-allelic segments help in pyramiding or pooling the best part of the QTLs together. And as well as I think you have to look at our paper that was published on the GSR breeding approach in the Theoretical Applied Genetics Journal recently in 2020. So I went out. Is that the question remaining?
So there are so many questions.
Okay. Yeah. I thought that was the end of the page actually.
No, there is so many. If when you feel it's okay, we can stop it. So
yeah, you let me know. But I based on the top priority on the top, I'm just telling them.
Okay.
But if it's similar question, I will if I had answered. But they're all different questions. That's very interesting for me too.
Yes.
The wild rice Rufi-Pogon cross can be a valuable pre-breeding material. Of course, any wild resources can be very useful. XA genes like XA23 came from Rufi-Pogon. Sometimes the yield genes have been cloned and from Rufi-Pogon. I think these are approaches where you can utilize these type of materials. Then David Chen puts a question, rightfully pointed out there are certain materials such as fertilizers, which are, originates from non-renewable resources. Assuming climate smart would be the ones that is most efficiently utilizing. Is there a quantitative measure of nitrogen efficiency between common varieties and climate smart varieties? I think this is a very important question. I think we published a recent paper in PLOS One, how this could be evaluated. And really, the phosphorus is one of the biggest challenges, because this is going to be completely, you'll not have a renewable source for this. And as we go along, spreading towards 2050, the challenge would become more serious as we go along. And these countries are located in Morocco, Russia, and all these countries. Whether they will trade these type of fertilizers even would be a question mark. How long they will keep mining these rock phosphates. And so these are really big questions. And of course, use efficiency, we actually use the partial factor productivity and the agronomic use efficiency for mapping the QTLs for that. But there are certain, the best approach would be the physiological use efficiency, which is very, very, physiological efficiency is one of the most important approaches to measure the differences between nitrogen use efficiency. And definitely the hybrids would stand out because of their heterotic combinations there. There will be much in higher use efficiency if we incorporate the genes rightly. Recently released hybrids and varieties in India, I think ICRRI has regular trials, and many of the companies put their hybrids there. I think one should refer to that. Sorry if I just skipped anything by mistake. Okay, Deepan, Roy, and Roy. Global Rice Research Array, HRDC doing the same thing for IRRI. Is it a question? Yeah, are Global Rice Research Array and HRDC doing? Yeah, kind of, because what eventually these type of arrays or these type of multi-location trials would be similar in some approach, but what we are trying to do in the new one rice breeding approach will be combining many of these things into one single way of testing these materials. And it will be very robust in its testing approach. And we are expecting very soon this will be laid out, and Hans is leading on this approach, and we will be seeing this happening soon. Sudhakar asked, nice presentation. Can these lines be accommodated to? Of course, these lines, many of these hybrids, as well as the inbreds that we developed are well adapted to aerobic conditions. And we have shared many of these lines with many countries in the past. How can we introduce two-line hybrid in our country? I think at this moment, two-line hybrids, we are sharing with the two-line study team, where we are trying to completely validate the commercialization of two-line hybrids from private sector point of view. And immediately these type of materials would be brought to different countries for testing. And the best way is to look out for this study group that has been formed recently in 2019. And this is a very, very robust set of companies, and public sector institutions have joined hands to see how this can be commercialized. I think once that is done, many of these things would eventually reach different countries. Do on-dries have anything to do with climate proofing? On-rise, to my understanding, it's a joke rather than a thing, that it is bird-proof, I believe. More than that, sometimes this on keeps these birds away. Sometimes that is the only thing, but generally on traits are not preferred in many parts of the world. But there are places where people like on also, because I told you it gives birds not damage it, because it gets it protects from the birds. So anyway, that is it also gets stuck up in their throat or something like that. So this is on rice, but I doubt whether it has any climate proofing. When we administrator, when we evaluate hybrids for yields or other characters, can we use for other commercial hybrid or check or standard varieties? When we evaluate hybrids for yields, okay, so we always use the hybrid checks and the best commercial market checks. So whenever the yields are comparable or bringing some new traits into them, or is robust in some manner, the people can easily evaluate it, and then based on the grain quality and the acceptance of the farmers, definitely one should be able to commercialize them. The next question is the anonymous attendee: what can we use hybrid rice in organic farming, because it need more quantity of nutrients, or so how to fulfill requirement of nutrients in organic farming? So of course, hybridized can be used in any type of farming. Farming approaches can vary, doesn't matter. And such hybrids, especially use efficient hybrids, should be a very perfect fit for this type of thing. But I would not encourage organic farming in the sense to keep food security when food security is becoming a challenge. Even if you put all the nutrients, they are not going to give you the best returns. And if you are going to use organic farming with organic fertilization and to replace the chemical fertilizers, I think that is going to be a challenge. But hybrids are always good in utilizing any kind of nutrients compared with inbreds. So it should not be a problem in adopting hybridized for organic farming. So you're welcome to use if it is your viewpoint. When we, okay, where are so many questions. Okay, so Arun asks, sir, whether photosensitive rice varieties can be converted to photoinsensitive, or any possible way to make it flowering? Yeah, of course, this is through mutation breeding. Of course, you can mutation or gene editing can be done, but GM is not in many plots of the world still. So you knock down this gene, you can make it insensitive. All those things can be done. I think it's possible. And through breeding also it is possible to replace the gene.
Sir, we think we received so many questions. There is 100 questions. We minimized half of them, and it's increasing. It's increasing like pop-up and increasing. So I think they are very much interested, and they understand, because there are so many comments in the YouTube that your every slide is very nice and very understandable. So that is the reason the question, number of questions. So, sir,
Yeah, I'm happy to answer, but I think time doesn't permit for everyone
yes,
to sit tightly. I think
yes. So
because
we wanted to move in our next session, that is the interview, because so many young fellow are waiting for that section. They are very appreciated how your career is built. So I'm giving the stage to Shoma. Shoma will conduct that interview.
Hello. We are now moving ahead to the interview section of today's webinar. Today is our 10th webinar of the series. We are honored to have with us Dr. Jauhar Ali from the International Rice Research Institute, Philippines. Sir, you're a pioneer in your field of research. We would love to know how you started your career. Please tell us about your journey so far as a plant scientist.
yeah, my journey actually was really very interesting. Right in the beginning, if I remember, as a child, actually, as a child of school-going kid, I had a deep interest in. We were located near to the IRRI, Indian Agriculture Research Institute. So very often I would go through those fields. And early on, I got this liking for agriculture. And I took a chance. After matriculation, I joined my BS Agriculture in Punjab Agriculture University, Ludhiana. There was a program to join after 10th. I said, why should I waste my 10th and 11th and 12th degree and then join, and I could save a year from that. So I joined after matriculation, and I saved 1 year of my time. Actually, it was a 5-year program. Then after joining there, I remember I had been very hugely influenced by stalwarts, rice stalwarts like M.S. Swaminathan, Dr. Gurdev Kush, Dr. Siddiq. They were actually in many ways, they were like mentors for me, everything. And I had the opportunity to listen to Dr. Kush. In the front rows, I was sitting as an undergraduate student of BS Agriculture in the front rows of the auditorium, listening to Dr. Kush when he was presenting the IR36 rice variety was being showcased. So in Punjab Agriculture University, I was in the front rows listening to him. That really moved me more towards crop breeding and genetics. And I took MS in genetics later at Indian Agriculture Research Institute, where had the opportunity at that time. I remember 1988, IRA introduced the entrance exam at that time. Many people may not be aware when IRA introduced the entrance exam. And to my good luck or bad luck, I don't know, I was interviewed by more than 400 people were interviewed for MS position for 7 slots of MS degree in genetics. And I was lucky to get through. I was a 4th ranker at that time. But anyway, so it was really exciting for me to get into IRI, and it's a very premier institute in this region, especially in India. And then I had the opportunity to work on chemical gameticides, or we call it as chemical hybridizing agents, for my master's. That was time when no CMS lines were available in India. Unlike, because this was a technology coming from China, then it came to IRRI, some of these materials, and IRRI was breeding these materials. And these IRRI bred CMS lines didn't enter India at that time in '88. And we worked on chemical hybridizing agents as an alternative approach to selectively sterilize the males and leaving the females fertile. And that way, can we produce chemically hybridized hybrids? So that was my MS thesis all about. And we discovered a group of chemicals called oxanolates. And that sparked a lot of interest at that time. And then I did my PhD also on 2-line hybrids. At that time, Maruyama in 1990 was one of the lead scientists who discovered the TGMS in Japonica background in northern PL12. And at the same time, in India, the scientists were looking how to get it done for Indian context. And at that time, a team went to Japan to Mariama's lab, and the materials were not to be shared with India at that time because of the huge costs. They were demanding a very huge money for that. So then the thesis problem came in IRRI, and they said, can we do repeat mutation breeding for that? I said, how can you search a needle in a haystack for hitting correctly that particular gene? But then we took our chances, and I did a mutation breeding with chemical and physical mutations. And I was lucky to get 8 TGMS lines we developed, and that was a very path-breaking effort. And it was our India's first approach where we could develop our own TGMS line in indica background for the first time. And this triggered the 2-line hybridized research in India, and for which I was awarded the Jawaharlal Nehru Award for outstanding PhD thesis. And then I joined Dr. S.K. Raina for my, who was also one of very great scientists on double haploids at that time, and he's still there in some private sector. And the double haploid breeding approach was a very good approach by which you can quickly fix the any kind of material segregating and reach the farmers much faster. So this learning from him helped me to speed up much of the breeding activities in the later part when I was trying to fix materials.
Okay.
Coming to, then I came to, I was selected for the 2-line hybridized postdoc at IRRI under Dr. Virmani. And Virmani is one of the stalwarts for hybridized, who was the first IRRI scientist who brought this technology to Asia and shared much of the materials initially from IRRI's germplasm to the members in different parts of the world. And IRRI could promote hybridized technology outside China in Asia specifically. And much of the work credit goes to Dr. Virmani, with whom I had a good association with him in that time when I was working for 2-line. And then I moved on from there to serve the Tamil Nadu Agriculture University at Agriculture College and Research Institute at Trichy, where I was trying to do salt-tolerant hybrids, which were we never thought of developing stress-tolerant hybrids at that early. Imagine sometime in '95, we had an ICR project grant from ad hoc project, and also NATP project also we got, and on 2-line hybridized project as well. So these projects helped me to develop the first salt-tolerant hybrid, especially sodicity-tolerant hybrid, CoRH2 from Tamil Nadu Agriculture University, which did very well in the sodic conditions where the pH is more than 9. And this hybrid did very well, more than 1.5 tons over the best Czech hybrid inbreds at that time. And then this journey from there, I came to, I was, after serving 5 years there, I came back to IRRI again for a second round of postdoc under the Rockefeller Foundation granted project, where I could work with a very lead scientist, genome specialist at that time was Dr. Zikang Li, and he's also was the GSR project leader as from Chinese Academy of Agricultural Sciences. And he was one of the very lead scientists with whom I had the opportunity to work with him as a postdoc in 2000 to 2003 as a project scientist at IRRI. And that is where I learned, I was the first generation of the molecular breeders from the Rockefeller Foundation, which invested on molecular breeders at that time. At that time, the technology was mostly on the SSR markers, moving away from the RAPD markers and all those markers, and came to SSR. SSR was considered as the best marker at that moment. And everybody working like day and night. I remember still many people working like shifts, night shift and day shift in the marker lab in IRRI. And this was a very exciting site, more than 15 postdocs in that lab called Genome Mapping Lab, GML, it was popularly called. And IRRI, then I, after this project scientist, I moved to a place where I was under IRRI Iran project. I moved to Iran for 6 years to lead their molecular breeding and hybrid program there. and I developed and helped in the development and deployment of Bahar 1, which is one of the very high yielding hybrids using the IRRI germplasm. And later also we developed about 6 hybrids, very high yielding hybrids for this country. And then I came to IRRI as the GSR project scientist, leading the GSR project at IRRI in 2009. And this was at a time when this whole thing was funded by Bill and Melinda Gates Foundation, and IRRI was a sub-grantee of Chinese Academy of Agriculture Sciences, and we were entrusted to develop and breed materials in the tropical context, whereas China is in temperate conditions, and it was a big challenge to adapt the materials coming from there. So we had to do a full-fledged breeding at IRRI to acclimatize and completely tropicalize the materials. And that's how the GSR breeding strategy evolved. And it helped us to map many of the traits related to many abiotic stresses. And that's this 10-year period from 2009 to 2019 was the most glorious period for me in my career, where I could, but my dream was fulfilled when I joined IRRI. To be honest with you all, that it was like joining IRRI was something like something like a dream come true for a kid when I was just thinking of just going to Punjab Agriculture University, and then from IRRI, and then coming to IRRI, and then becoming a scientist at IRRI was really a very pleasant moment for me. And really, I feel that the opportunity that IRRI gave me to unleash my potential is very much acknowledged. And I feel for all youngsters that they should always aspire to come to IRRI and get connected with us. This is a very good opportunity for everyone. So I could release about 28 varieties directly bred by me in less than 7 to 8 years, and reaching 2.7 million hectares and almost 2.2 million farmers touched. 110 of these materials are in the pipeline in different countries to be released. So how productive you can be beyond this, I don't realize. And now with the hybrids that we are developing, we have hundreds of hybrids in the pipeline that will hit the commercialization wing very soon. And this is what is the potential of these materials will be unleashed as we go along. So this is where my journey of leading the hybridized program at IRRI, where we revitalized the hybridized development consortium. And today we have 88 members from public and private sector. And this is a very good platform where the public and private sectors come together and learn a lot of things, where we share our materials with this platform. And we get nominal fees from them, and a nominal licensing fees that goes back into research again of IRRI's mandate to serve the poor farmers in the world, to reduce the costs and all those things. I think I don't want to extend more than this. I think this is where the journey ends, and it's still very bright for me to move along. And some other time I will tell you the rest part of the story as we go along. Thank you.
Sir. Sir, how important a role will hybrid rice play in feeding the billions in the future?
You see, the hybrid rice is such a wonderful technology. You have to understand the core of the issue here. What is your parents is what is your F1. If you design your parents according to the requirements of the market segments, and definitely the hybrids will perform the same way. And if you have very good system of market segmentation and understanding market requirements and breeding the right parental lines for different, definitely the hybrids can give 25 to 30% yield. Now, 25 to 30% yield means 25 to 30% resources you are saving. Like suppose you have 100 hectares of land of inbreds, and I want to put in terms of production, if I just have to use hybrids, I will get it done in 70 hectares, the same yield. So 30 hectares is saved. In just to understand in a layman's language, that 30 hectares of inputs is saved, 30% of the labor is saved, 30% of the inputs that is very costly these days are being saved. So hybrid technology does so much in that front. Then you have the second layer of it gives employment generation to the industry. It also helps the farmers to come out of this the so-called 1 ton means the marginal advantages that they get from inbred technologies. Of course, inbred technology is important, but it has to be utilized in places where the manifestation of the heterosis is maximized. Where you get 25 to 30%, definitely one should exploit. So when you translate this into a larger area, now 8 million hectares outside China is hybridized. If we can touch 15 million hectares in the next 10 years, I'm sure that we can meet the challenge of 2050 to feed globally, especially who are depending on rice as their staple diet. I think I will stop here.
Sir, along with food security, what are your thoughts on nutritional security of rice?
Yeah, this is a very important topic. I think it's a very important thing that the second dimension of food security has to go along with the nutritional security. And I also mentioned in my first slide, when I mentioned that to for the viewers, that this nutritional security is so vital that it's it is not just food security is just not just solving the hunger, but the hidden hunger is solved by the nutritional security. There are a lot of hidden hunger in terms of zinc requirements, dietary requirements of iron, zinc, and many other elements that is missing in many of our diets. And by putting these things in right perspective and making a mainstreaming of breeding methodologies by which we incorporate zinc and iron into our all breeding pipelines, this can be resolved. And very easily it can be done. It's just some few major genes that is required to be brought into the breeding pipelines, where we can use them even in the forward breeding approach, 1 or 2 markers for that to select automatically those materials, and we can forward it, and very easily it can be done. And anybody can do it. It's not some lead institution doing it. Any breeder with that mindset should be able to involve the zinc and iron to be incorporated in their breeding programs. But one thing, one caution here is one has to be very careful when you are using this type of heavy metals like zinc specifically. They have a lot of interaction with other heavy metals as well. So in places where arsenic and cadmium, they have huge interactions and interplays between these type of heavy metals. So one should not put them, their uptake mechanisms are similar. So one has to be very careful in breeding zinc-rich materials, but not to be deployed in places where arsenic is a problem. So we have to be, deployment has to be very careful. So therefore, the market segment analysis is very important before deployment strategies now. So this is the future, very important for nutritional security. Yeah.
Okay. Sir, what new tools and techniques will be important for rice breeding in the future?
I think this is a very important, we are evolving actually from simple marker-assisted breeding in the past, and simple breeding, marker-assisted was considered as a big tool at that time. And then we came on to the whole genome sequences were done. And now when all the molecular markers and high density maps are available, we know more information on the genotype of the materials that we are breeding. We can play with these, the whole genome sequences to our advantage, where we can easily pool them, paramate them, and do many things, including the genomic selection nowadays is the buzzword. I think genomic selection by using elite by elite founder lines certainly should help in advancing the genetic gains much rapidly. But for the youngsters, I see that these the new tools mostly related to computers and related to gene sequencing should not tie them up with the computers and servers and running after the data, but without looking at the plants in the field. My breeding always came from the field evaluation and selection, and then fully enforced with the genomic knowledge of the materials and their how they move in the structure. and then analyzed them and using design QTL pyramiding. So the GSR breeding technology, the design QTL pyramiding approaches, and the rapid generation cycles to maximize your genetic gains. We call nowadays speed breeding, and a lot of good publications are there where you can do 6 seasons in a year in barley and other crops. For rice, still it is 3.5 crops you can take in a year, but that's a challenge. But this can really rapidly, you can advance your generations and genetic gains can be maximized. And above that, if you use some of the best breeding strategies like genomic selection, GSR breeding strategies, design QTL pyramiding, I think these can do wonders for any breeding program. And this can be extended to not only rice, any crop. Don't get restricted. This is open to any crop, and these are most of the crops are being used by our sister CG centers as well.
Okay, sir. Now we'll conclude with one last question. What would your words of advice be to the young research fellows out there?
I think the words of advice, I'm not in a position to give advice to anyone, but some of the youngsters, I think what I would suggest them is there is no replacement to hard work. No matter you do anything, you have to put all your dedication, your efforts, and these are proven pathways already. It's not that we are going for the first time learning something. You have to use the proven pathways of people who have experience, and I'm telling you that it was not an easy journey for me to reach here. And if you have to do, it was sheer hard work, and true dedication to see that your materials reach the farmers. So there should be some kind of a driving force or passion to drive you exactly. So sometimes this passion drives you to be dedicated, and you always remember that, look at that poor farmer there in somewhere your materials will be utilized. Can that bring smiles to his faces? That is what drives me personally. And that motivates me every day. Okay, somebody is using my material and trying to benefit by even half a ton in the best conditions. It's good that gives a lot of rewarding, self-rewarding yourself, that somebody is benefited out of it. And then apart from this, some of the students, they have to understand that it's not, they have to put very dedicated efforts to get to the bottom of it, basically. Like, you should be passionate, you should have inquisitive mindset, you should try to go to the bottom of everything, not just look at the top and just do something and superficially touch it, but get to the bottom of it. And this is very important for youngsters, especially in the jet set age of modern computers and gadgets and media and all those things. So they get often diverted. So they should go deeper. A deeper dive is very essential for science. And unless you get satisfied personally, I don't think no better rewarding than your self-satisfaction and what you're doing at the end of the day. I think I like to just put this as a prick to the budding scientists, so that they get started right away. Thank you.
Okay, thank you so much, sir. We will now go back to Shubho.
Thank you, sir. I just wanted to show you our today's responses, because it's very hard to calculate the viewers. It's easy to calculate that we got within 4 seconds 500 attendees in the Zoom box, and I noted about 955 in YouTube, but it is varied. So the best way how we understand that what is the today's attendees, I want to share the screen to you, and this is ongoing. We got 1,928 responses, and from these countries we got 1,674 responses from India, from Nepal, Philippines, Pakistan, Bangladesh, Indonesia, Malaysia, Nigeria, China, Morocco, Tunisia, Algeria, Australia, Cambodia, Egypt, Iraq, Saudi Arabia, Singapore, Sri Lanka, Thailand, Vietnam, Benin, Bhutan, Ghana, Ireland, Kenya, Mexico, Oman, Peru, Sudan, Zimbabwe, and many more. So I think uh, it is the reward, and it is the satisfaction to you, and for us also, that so many people are viewing today's talk, and so many questions. I don't know, there is a lot of questions. So many private sectors company from Pakistan, they are going to approach with you about your work. And I think this work, this webinar is fruitful to me. I am expecting such responses, and I'm very thankful to you.
I think one should congratulate you and your team actually for this wonderful. I'm very delighted to that so many people listen to my talk, and certainly it's a very humble request that I will try to answer many of these questions at a later date. If I get their email IDs or something, I can respond to them as well. At least some of these questions, if you can post me back at IRRI, we can put a staff for sorting out these questions, and we can send back as feedback to you, which you can post back into your YouTube or somewhere, so that I don't want the questions get lost somewhere in this huge responses that came. Certainly, we'll try to respond to every question and put it in YouTube section, which I will share with Shubho later on. I promise that. Yeah. And I also thank all the viewers for patiently bearing with me. Sometimes I get excited and I think I didn't cross the time, but I hope you enjoyed my presentation. Thank you.
Thank you, sir. And we have we have the feedback form and all the email address of the actual attendees, so we can forward your answer to them any point of time in the future. And we are going to share, without their disclosing their email address and phone number, we are going to share their institute, their profession, and their name in our website, and we allot their certificate number. So I also share you the link to tonight. You can see the actual professions wise everything. So it's so wonderful being with you. I really, I don't expect that you agree to deliver a talk, but it motivates me a lot. Even your presence give me so many positivity for your talks, that I can move forward. We can all together, if it is for the benefit of plant science, and we are all plant loving people. So it is thankful, and thanks from my team and from my.
From my side and IRRI side, I think I'd like to thank the team Bioengine and the panelists out here. They are doing a wonderful job, and I'm sure this platform will grow to strength and with time, and keep doing this effort. And certainly there will be more instances that more learned people will come and talk in your platform, and it would encourage science to progress. This is exactly what IRRI is all about. IRRI wants to share knowledge with its partners, and people who look towards IRRI, they are welcome to IRRI, and look at our website and look at our thing, and get connected with us. Thank you very much.
Thank you, sir. We have some few minutes we wanted to share with our viewers. You can, sir, you leave the meeting. You can leave the meeting.
Thank you. Thank you very much.
Thank you, sir. Bye-bye.
Bye.
Now I hand it over to Shoma, give some instruction regarding the certificates, because today we got so many new viewers also. So it's maybe their answers to how to get the certificate and what is the next. Today we got so many suggestions of future topic. I'm assuring all of you, we try our best, but we need the speaker also. Those are the expert on that topic. I jot down, and with the help of our two volunteers. So we we try to communicate the respected expert on that field. If it is possible, definitely in near future, it will be available, and we are happy to host those topics and those speakers in this platform. We already set and start the registration for the webinar in August. There are 4 webinars because we try to lower down the number of webinars, because in July we are arrange 11 webinars. So in future we try to 2 to 4 webinars in a month, which gives us the time to giving the certificate, to inquiries, to arrange. And we also have our own work. We are all a researcher, an active researcher, so we need some time to our own work also. So hope you guys are understand that so many webinars is not possible, but we planned in such a way that many talks can possible, and many of you are learn, and I also very interested in this talk also, in this on the future talks. Soma will help to understand our future talks and available options which we gave under the Bioengine to Shoma.
Okay, I'll share my screen now. Okay. Is it visible? Oh, okay. Wait. Okay. This is the first one. Is it visible? Okay. So you can see that there is the password for today's feedback form, which is well, you need it for the certificates. Regarding the certificates, you can apply for the certificate for attendance for today's webinar. We give out certificates of attendance, and if you have attended the webinar, you can apply for the certificate. All you need to do is you need to click on the feedback link, which is you can see it in the chat box in Zoom and YouTube. If you don't find the feedback link, you can also go to our website. There is the feedback link is also present there. You will need the password, which is this is the password on the screen, and also it is there in the chat box. Just get the password, because there is a place when you need to type the password in the feedback form. You fill out the feedback form, and then you save a copy of the Google response sheet, because you may need it later. After filling out the form, you will find that you can save a response sheet. Okay. Another thing I want to say is that you only have 2 hours time for this filling out of the form. The link will be active till 2:10 Indian Standard Time. So within 2 hours it will close. Also, regarding the certificates, we are not sending the certificates individually by email. So what we are doing is we take 2 days for this to make this happen. We are receiving all the applications from you through the feedback form. After we receive all the applications, we will make a list with a file number. So you get this list from our website. Go to our website homepage, scroll down, and you will find all the previous webinars have been listed there with the name of the topic. You click on the topic name, it is a hyperlink. You click on the topic name and you will see that an Excel sheet opens up. You will find all the attendees, the list of all the attendees who attended today's webinar. You can find that there's a name and there's a file number. So you click, you find out your name. It's all arranged alphabetically. You find out your name and you will be allotted a file number. Then you go back to the home page, and then you see that then where it is written that title of the webinar, there a certificate is written in hyperlink. You click on the certificate hyperlink, and you will go straight into the Google Drive folder. We have shared the certificate folder, the Google Drive folder. You get in the folder and give it some time to load, because it is a big folder, so it will take some time to load. And you need a good internet speed. Then you can see that all the PDFs are there with the file numbers. So you have your file number, and you download your PDF certificate. That's very easy. And also you don't have to wait for anybody. You just have to wait for 2 days after every webinar, so that we can do this. We can make the certificates and make the sheet and put it in the drive and share the link. Okay, so we need 2 days for this, because many people are asking right after the webinar, or today evening they're asking, where is my certificate? So we need 2 days. Please give us 2 days time for this. Go to our website after 2 days, and you will find that the title name and the certificate link is there. Click on the title name, get your PDF file number, click on the certificate link, download your certificate. Okay. Next, this is our next webinar. It is on 24th July. The speaker is Dr. Raviraj Banakar. He recently joined Avastagen, and he came back to India from University of Minnesota, USA recently, and joined Avastagen. He will be talking on the topic CRISPR-Cas precision genome engineering. The registrations have been open for a long time now and are still open. Please register for this. You can visit our website. Every webinar details are there of all the upcoming webinars. You will find the detail about this webinar also there. You can click on the registration link and register for this webinar. Okay. And these are the webinars in the month of August. For the month of August, we have arranged 4 webinars. The first one is on 6th August, then 11th August, then 17th August, then on 24th August. You can find all the details on our website, and the registration links are also open. You can go in and register. Okay, while you're registering, or while you are filling out any type of form, registration form or form of certificate application, you are inputting some data like your name, your email address. I just want to request some of you that some of you mistype your email address. So please see that you have typed in your email address correctly, because if you type it incorrectly, then you will not receive any mail from our end because of the wrong email address. Okay, so please check that your email address is correct. I have to check, like for today's registration, there were almost 5,000, over 5,000 registrations. I had to go through each and every one of them to. And one other thing I wanted to say is that please don't share your email address with anyone while registering. One name, one email address. Okay. If one person, if there are 2 people with the same email address, I don't know how we will sort it out. It is not good, because it's not fair either, because one person can apply for so many uncountable number of people. So no, that is not happening here. One person will have one email address, only one email address. Okay, don't share with anyone for the purpose of registration. You make your own email address. Okay, regarding the name on the certificate, write in your name correctly. First of all, type it correctly, because I have been receiving mistakes on your name, and then you are requesting by mail that please, I have made a mistake in typing my name, or I have forgotten to write my middle name. Please write in my name correctly. So for so many people, correcting their names will be very difficult for us. Please write it correctly. Also, don't put in your designation in your certificate application form, just write in your name. Okay, that's it from me. That's all I had to say. See you again on 24th July for the next webinar. Thank you. Okay. Stop screen sharing. How do I stop the screen sharing?
Thank you, Shoma.
Okay, wait.
Now we are at the end of today's webinar. I'm personally request all of you, because all the email I received, and actually I give them the feedback, and I'm feel very unhappy when I not giving the answer, because I like to give the answer of your email, but it is very hectic to unnecessary emailing me, because our rules is rules, and we can't change the rules, because it is a new platform and rules make us the better. So asking for the password, asking for the feedback link after the end of this webinar is not going to be answered. Your internet problem, your locality, your personal reason is not going to be entertained, and we are not giving you such opportunity, and we are not giving you the feedback link and password later the 2:20 or just ending the webinar session. You have to come in the live webinar, listen, learn, because it's all free. But we also organized it because we can't give password, email address before or after the webinar. So I'm requesting all of you, don't mail us on such issues, or we are not going to answer your mail. Second is, when I upload the certificate, I carefully check all the certificates are there or not, because when I upload it, automatically Google Drive folder shows me that this number of certificate has been uploaded. So there is no ambiguity, there is no confusion that whether certificate is not there or not. If it is not there, check it, check check with a high internet speed. So I think all the certificate are there. I'm after assuring I shared the drive, so there is no chance that your certificate is not there. It will be happen that your name is not on the list because you have not submitted your feedback, because it's the auto-generated list. It is auto-generated list, and it's automatically sorted out by the Google Forms. So if you able to submit your feedback, whatever the reason, if you not able to submit your feedback, whatever the reason, you will not get the certificate. So if you submitted your feedback form, you will get the certificate, but we need to scrutiny your form, because we wanted to your name, not your designation, not your institute, and not your profession, current profession, because these are the different form. So many people are put their name with their designation we in the certificate application form, because in every certificate I issued, my name is mentioned there, and it's my responsibility to check all the certificate before I'm going to issue them. So I need this scrutiny time. We need 2 days. After 2 days, you come and collect your certificate from our website. If you're not getting your certificate, but your name is on the list, check multiple times with high-speed internet, using the computer, using the Google Chrome, logging into the Google Drive or Google account. It has to be there. Don't email us on that basis. If your name not in the list, this is the reason that you not giving the feedback form. And we can't include your name after 2:10 p.m. of today's webinar. So best of luck. Do your feedback, and please come at early to listen the talk. Some guys have come into at the end. I got just a request, feedback link, please. What the meaning of that? The webinar is ended. It's now somebody asking for what is the link, please. Don't ask such type of, don't ask such, don't, what I say, don't, it's not a good approach to listen that type of webinar. Our all webinar, I think we give enough time to listen and learn, not that 20 minutes or 25 minutes where the webinar is for a good time, good explanation you got. So it's a good learning process. I also learned from this webinar a lot. So this is the end of today's webinar. As I got the request, I just put the last time today's password and the link, but please come at 11. We start at the sharp time. Come at the 11. Listen for something from our webinar series. See you guys in 24. There is a talk on CRISPR. Thank you. And all the related information you guys are able to find it from our website, as our website is not built yet, but you can go for bioengine.com. There is a mention a link. There is our alternative Google Slides drive. So I suggest if you have any question, any query, mail us. But it's a very easy process to listen Bioengine webinar and apply the feedback form. Thank you. Thank you, everyone. I'm going to stop this webinar, and this video will be available very soon. Due to back-to-back webinar, I'm unable to put all the videos, but I think within 2 days, I'll able to put all the videos in our YouTube channel. Please subscribe our YouTube channel. It will help us to communicate you in a better way. Thank you.